Hello, and welcome to IB TutorVids. I'm Jordan Dermaz, and this is How Do Repressors Work? Now this was a question that was asked to me right before the AP Biology exam, and there's a study guide resource on my website explaining the process. But today I want to just go over the acronym and a little more into depth what does the process do and what does it mean for prokaryotes. Now here I have the acronym for the 4 different types of genes you'll find in prokaryotes. One way to remember just all 4 of them is PORS, because it's a very easy word to remember and a very common word. Now PORS will stand for promoter, operator, regulator, RE, and structural genes. However, to have them in the correct order of process and operation, it could be REPOS, R-E-P-O-S, which is regulator gene, promoter, operator, and structural genes. Now remember that it's structural genes, because there are multiple genes within a operon. Now an operon consists of 3 different types of genes: the promoter region, the operator region, and the structural genes. The regulator gene and the prokaryotes can be anywhere in the DNA of the prokaryote, and it can code for multiple operators. Now this regulator gene, as we know, genes when transcribed and translated create a protein. Now this protein binds to the operator and turns it on or off, almost like an on and off switch. And this can change what structural genes get coded. And what will happen is the DNA polymerase, the RNA polymerase coding for this gene, or the structural genes, will bind to the promoter region and try to code this, but get to the operator and not be able to. Because the regulator creates a repressor. The repressor blocks the operator and inhibits transcription. Now we see it can also inhibit certain type. So for example, it can make it not code for structural gene 2, but only code for structural gene 1. This process is mainly applied in the IB and AP exam with the lac operon, and it's called the lac operon because it codes for genes within the prokaryote that digest lactose. Now, the regulator gene also has a little tiny promoter region, and when lactose comes into a prokaryote, it binds to this region, which codes for the regulator, which releases the operator, which stops the inhibition of the operator. Or, the way the process is usually explained is that the inhibitor has an active site on the operator, and the lactose fits right into the active site, which causes, as we know, when a protein or when an enzyme is activated, it changes shape, and the shape no longer inhibits the operator, and it's released, and now it can be coded for digestion of lactose. Now, because prokaryotes are not usually exposed to lactose, they don't usually need this, so the regulator usually blocks the operator so they don't have unnecessary proteins. But once they come in contact with lactose, this is freed and coding can begin, and multiple proteins can be made. An RNA polymerase can come here, can come here, can come here again, and code multiple RNA strands, and those multiple RNA strands can have multiple ribosomes on it, translating and making a polypeptide. Now, the most important part of this to remember is that this is only in prokaryotes. In eukaryotes, the process is completely different. Sure, there's something similar to a regulator, and there's a little tiny promoter region where the DNA or RNA polymerase binds, but you don't have the operator and that type of regulation. Now, another thing is that the process can be more complicated. Once there's too many enzymes, it's called end product inhibition. The end product, which is the enzymes, can stop the coding of, or start rather, the coding of the regulator gene, which stops the operator. And the last thing to remember is, and I've already mentioned this multiple times, is that there's many structural genes. So one of the most common questions on the AP Biology exam is they'll give you multiple choices and which one looks right for a prokaryote. And it will show you a picture that looks like this, They'll show you a picture with one structural gene and another structural gene over there. They'll show the regulator right up against this and the promoter over there. All you gotta know is this order. And remember that this is plural, and remember this doesn't have to be with the operon. And then you're golden. Also, always think of, it's very easy to think of an enzyme as a lock and key. That's one of the easiest ways to think about it is a chemical is the key and the lock is the enzyme. And the chemical fits into the enzyme, the substrate fits into the enzyme and changes it so it opens the lock and causes it to do something. If you're needed to explain the process in, for example, a long essay question for the AP Biology exam or an FRQ for the IB Biology exam, that is very important that you understand the process of the enzymes. For more information, you can visit my website at tudovids.weebly.com or email me at ibtudovids@gmail.com. Now remember, there is a resource online that goes into detail about this, as I did, and goes over the steps and includes pictures and images.